use and you can actually start looking at the internal structure of the cell, what's happening inside, does it have symbionts and so on. Okay, now I have another.
You're all good. It's just your mic.
Okay, I'm gonna completely freak out. I didn't have enough sleep, but or general. Okay, so we can, some other pigments fluoresce. Lots of organic material in the ocean fluoresce. The stuff that we call sedum, not everything fluoresces, but quite a lot of sedum fluoresces. Oil, like oil spills and stuff, oil fluoresces. There's certain minerals that also fluoresce, but never probably enough for us to actually make a meaningful measurement around it. But what is fluorescence? Do you guys remember from what Meg was talking about, what fluorescence is? Anybody? Idea? Anybody been to a club? Had time? Yeah, exactly. So I'm a molecule, whatever. I absorb light, a certain energy. I'm gonna use a little bit in thermal, and I'm gonna shoot that light now, but I lost some of the energy, so I'm gonna shoot in a lower energy. If I lose the energy, there's a preservation of the pain and light field, and so I have to move a little bit towards the red in a wavelength spectrum. So like you're absorbing a light of a certain energy, a certain wavelength, and you're shooting out a photon of a lower energy and a higher wavelength. I'm sure people have noticed it before, but Stokes was the guy who actually described it in physical terms. So we call the shift Stokes shift, or it's easy to remember a red shift. You're going towards the red, and that's pretty much what fluorescence is, in very, very simplistic terms. This fluorescence usually is specific to the molecule itself and has a unique fingerprint, meaning that specific absorption is going to give you a specific emission of the fluorescence. So the wavelength and energy are going to be highly specific for a specific molecule that we are evaluating. You've seen this in Tuesday. Meg went really deeply through Jablonski's diagrams, and I'm not a chemical or, my physical chemistry was not my strong point, but you guys know that there has to be a certain specific amount of energy in order to excite molecule or electron to get it from S0, from the ground state to first excited state, or any kind of excited state higher up. Fluorescence can only occur when the energy is released from the first excited state to the ground state. It can't happen from S2 to S1. It always has to happen from S1 to S0, or ground state, whatever we call it. To look at the phosphorescence, we're not going to talk about that stuff, but just fluorescence, just remember, it has to go from S1 to the ground state. When you think about the processes and the timeline processes in the molecule, absorption is a relatively fast process, and I think Meg mentioned that stuff. It happens really super fast. We're talking about the order of 10 to the minus 15th of a second, Fluorescence slows that down. There's magic happening there. Some of the energy is getting lost to thermal. And in theory, photon forgets where it was coming from, and that process takes some time. Then fluorescence takes some time. So it's a little bit slower than the absorption itself, but it's still super fast. And that's when fluorescence is happening in a molecule. We're still sitting in a little molecule, chlorophyll or a specific acetyl molecule or so on. So that's your basic fluorescence. So you want to remember that, energy gets absorbed on a certain wavelength. Photon gets absorbed of a certain energy, certain wavelength. Some of that energy gets lost was thermal or somewhere else, but thermal. And then it gets emitted of a lower energy, higher wavelength, and moving towards the red. It can only happen for the specific molecules that are capable of fluorescing, and it can only happen from the first excitation state back to the ground state. And just your fluorescence. Really basic. This is biologist view of fluorescence. If you want to know more, talk to Colin. I have put a link to Colin's talk from 2 years ago, where she really goes to really beautiful details about that. And you really want to, if you want to know everything about the vibrational states and everything what is happening, go there. What fluorescence, as I said previously, chlorophyll, color-dissolved organic matter, oil, phycoerythrin, some minerals. Some of these measurements are useful, some of these measurements are not useful. Some of this fluorescence we can see from space. So here you have an image of naturally induced, sun-induced fluorescence that we can actually see from space of the chlorophyll. And you can see the distribution of this, higher values. So greens and yellows and stuff are kind of patchy, and usually, it's sitting in the areas where we would expect higher chlorophyll concentration. So along the shores and the highly productive areas, let's say of North Atlantic. Now, we can use this measurement fluorescence, so measurements of the inherent optical property, because fluorescence is type of scattering, inelastic scattering, as a proxy for biogeochemical parameters of interest. And chlorophyll fluorescence can be used as proxy for the chlorophyll concentration. I'm going to talk about that. For phytoplankton biomass, because phytoplankton has chlorophyll, as we heard from Sasha. Since we know that phytoplankton, takes up the light, and through the process of photosynthesis produces carbon, probably we can say something about photosynthesis as well. We can also say something about physiological state of the cell. And I'm going to just touch a little bit based on a little bit on that. Phycoerythrin fluorescence, that's the one that I showed that is usually associated with cyanobacteria. You can also use it as a proxy for phycoerythrin concentration, but you can also use it for cyanobacterial biomass. And I think Sasha covered this really well when she was talking about usage of a phycoerythrin fluorescence in flow cytometry to distinguish different types of population of the teeny tiny picoplanktonic cells. So Cetinić was in this situation the only one that had phycoerythrin. So if we plot the red fluorescence that is present in all these cells versus orange fluorescence, Cetinić is going to pop up. So we can use this fluorescence as a tool to distinguish different type of populations, say something about concentration of Cetinić in the water. And ultimately, we have CDOM fluorescence. And CDOM fluorescence, it's a little bit more complicated. I'm going to touch a little bit on it. And it can be used as a proxy for CDOM concentration or absorption. Or in really very, very particular situations, you can connect the CDOM fluorescence as a proxy for dissolved organic carbon concentration. As you're going to see, and I'm going to touch a little bit on it, CDOM is, like you say, plants now, everything. It's such a diverse pool of organic molecules that have their own specific optical response, its own specific fluorescence. Not all dissolved organic carbon fluoresces. So if we're going to use CDOM fluorescence as a proxy for dissolved organic carbon, you have to be very, very focused area. You have to know a lot about your system and have lots of samples to actually build a really strong relationship. But don't collect some samples here in Hartsville Sound and apply it globally. That's not going to work. Fluorescence can be measured in many different ways. We can do fluorescence excitation spectrum, which means that we can monitor fluorescence in certain wavelengths, let's say 675, and emit the light starting from 400, for example, all the way to that wavelength. And then we can observe the response, the fluorescent intensity as a function of that excitation light. So we start, we have a molecule, 300, 350, 400, and the other side, the only thing we're measuring is light at 675. So we're looking at the response of the fluorescence as a function of the changing light. So we can say something about the way the molecule fluoresces based on the excitation wavelengths. So I can go in details, I will not. Second thing, we can flip it around and we can look at the fluorescence emission spectrum. So we can excite the molecule in single wavelengths and then measure the fluorescence in all the other wavelengths. So you excite the molecule for 400 nanometers and you start measuring the emission 400, let's say all the way to 750, which is going to tell you a little bit how that energy is like propagating, where is it going to be fluorescing later on. You can combine these two, and I'm going to show you really fast this fluorescence excitation emission plots, which are very often used in analysis of dissolved organic materials, CDOM fluorescence, and it can be used to kind of say something about the complexity or different types of CDOM that are in your water. Fluorescence emission spectrum, in theory, it's a combined thing in a sense, because what you see from space, the natural fluorescence, in a sense, is also it's a combination, because the sunlight that is shooting at the ocean, it's a broad band, and what you're measuring, the reflectance, is a broad band too. But you're not going to mean, again, there's absorption, there's other things happening here, so you're going to be kind of looking for that fluorescence in a specific portion of your ocean color that you're receiving on a satellite. But it is kind of like fluorescence excitation emission spectrum in a really bad way. And then you can do single excitation, single emission. And that's usually what we do. This is what you guys are going to be doing today. You're going to excite the molecule at a specific wavelength, and you're going to look at the response, the emission at a specific wavelength. And it's something that is done the most because it's a simple instrument that you can just take on a field. You can't something that is like emission excitation spectrum. You really need an instrument. I mean, Colin has one. It's something like a spectrophotometer. It's very optically, you need to work with it a lot. I have some data from some field versions of that instrument, but usually when you do fluorescence, you usually use single excitation and emission. There's certain models of maybe you can measure different emissions of different excitations to distinguish different phytoplankton types, but usually a single emission, single excitation sensor. And that's what you guys are going to be doing today to measure chlorophyll and a little bit of acetum. Okay, so really fast, acetum fluorescence. It's, as I said, this is a really complex thing. When you say acetum, it's like you say all the plants. Now, there's so much diversity in there. And here it's an example of a study done somewhere, I'm going to say somewhere in Germany, based on the last name, where they really looked at a bunch of different organic molecules they defined in acetum, and they did excitation, emission, absorption spectra. And you can just look at different types of curves that you can see here. So this is like, okay, if I see this correctly, so this would be excitation. Red line is absorption, and the dashed line would be emission spectra. So you can see how diverse they are. There's a gigantic range of these excitation and emissions and fluorescence spectra. So, and we tend to dump them all in the same thing. It's really hard to do. So what people are doing, and I really like that a lot, is they're measuring this excitation emission spectra. So what you do, once again, you started your excitation here, and you're shooting the light, and you're measuring your response and all the wavelengths, And as you're doing, you get this really beautiful three-dimensional idea of the specific response of the molecules that you encountered. And then you can do like analysis similar to the one that Sasha was doing, where you're actually decomposing that stuff using some kind of statistical approach. Sasha was talking about the Gaussian decomposition. This is something done similar to PCA, so primary component analysis, where they just decompose the modes of the behavior. And by taking that in consideration, they have managed to distinguish three main modes, but they can separate terrestrial sources of CDOM, something that's more marine, and something that is more protein-like associated with the phytoplankton production. Pretty cool. But now we're going to go to chlorophyll, because this is something that I like a lot. It pays my bills, and I have it every Christmas on my tree as a molecule of chlorophyll because, again, I love it, pays my bills, I love it. So this is the molecule chlorophyll. I know, have you seen it before? This is how it looks like. It has this porphyrin, I can't speak. There's a ring up there in the middle of which the magnesium sits, and it has this really cool tail, no? And the only difference between chlorophyll A, that we talk about a lot, that is present in mostly all the phytoplankton, and chlorophyll B is this little thing up here on the top. In cells, we can also find, except for chlorophyll A and chlorophyll B, some other chlorophylls, and we can very often find degradation pigments that also have a fluorescence signal to them. And we're going to be measuring that today in order to distinguish chlorophyll from these degradation pigments. Chlorophyll A fluoresces in solvent. Chlorophyll B fluoresces in solvent. So once we extract it, chlorophyll B does not fluoresce in cell. Only chlorophyll A fluoresces in cell. Some of these degraded pigments that fluoresce in solvent, and they can cause some little problems as you're measuring chlorophyll inside of the solvent, and you're trying to connect this chlorophyll fluorescence to chlorophyll concentration. Okay, so this is something where I'm going to point you out to check the Collins lecture, where she really goes beautifully in the whole idea of absorption, to this S2 level, going down through all these vibrational levels, losing this absorbed energy to the heat, and how is that secondary absorption relating to the fluorescence. I'm not going to talk about that stuff. I'm going to mess up something. Check Colin's lecture. It's LinkedIn, so just click on it. It's gorgeous. Look at that stuff. But the basic idea that I want you to take home is that chlorophyll fluorescence is related to the absorption, which we know because the light has to be absorbed. So we have, if we want to say something about fluorescence, we have to know something about, that's not working anymore, absorption of the chlorophyll. We have to know something about the energy that has been used to excite that molecule. And then we have to know something about this term that looks funny, but it's called quantum yield of fluorescence. And that's actually a term that tells you that it's pretty much a ratio of the energy absorbed and an energy fluoresced. It just tells you all the energy that was being absorbed, how much is it going to go out on the other side, no? So, you know, you're measuring chlorophyll fluorescence at one wavelength because it has to be a little bit down the line from the excitation wavelength, lambda 1. And the quantum yield of fluorescence is specific to those two wavelengths, you know? It's, if you change the wavelengths, the quantum yield, so the capability to fluoresce what it was absorbed is going to change. And so if you now take that chlorophyll and rip it out of the cell, and you guys remember when we did the methanol extraction, we killed those cells, we destroyed those cells, we diluted those cells, pretty much dissolved the cells so the pigment can go away. When you're actually measuring pigments, you can capture that organic solvent with the pigments inside. And that's what we do when we're trying to use the chlorophyll fluorescence in order to relate it to the concentration of chlorophyll. So in that situation, chlorophyll molecules are in solution, no? We have a constant mass specific absorption because we know that it's not going to change. It's just a molecular chlorophyll. The constant quantum yield, because in this case, quantum yield is just related to the molecule. We know exactly how much the molecule will absorb and how much will it fluoresce in a specific solvent on a specific temperature with a specific pH. So we, for example, for the chlorophyll in acetone, the quantum yield of fluorescence is around 0.3. If you move to a different solvent, you might have to go up to 2 different wavelengths, and maybe the quantum yield will change a little bit. But we're going to be doing it in acetone. If you maintain constant energy, and you can do that with a really good light source, which we have really stable lamps usually, in that situation, fluorescence of the chlorophyll is equal to concentration of chlorophyll in solvent. Okay, because you don't have any problems to deal with. How do you solve that? You take your chlorofluorometer, you make a dilution series of a known chlorophyll concentration, and you just measure, you know how much chlorophyll you have, and you measure your fluorescence. You know how much chlorophyll you have, you measure your fluorescence. You do like a dilution series usually of 9 or 10 dilution points. And once you have this relationship, you have this way to translate chlorophyll fluorescence to chlorophyll. It's super awesome. It's super easy. It's super cheap. Every measurement of chlorophyll fluorescence, every measurement of chlorophyll using this approach costs you around $2 or $3. HPLC, which is our golden standards, cost around $130. So now the question is that chlorophyll fluorescent-based measurement of chlorophyll concentration good enough? There's a little study that we just did in a lab, and Amy is trying to publish it, and she actually finds really good correlation between HPLC measured chlorophyll and chlorophyll A measured by the fluorescent approach. So without going into statistics, everything that is within green is good. I'm not going to explain the statistics here. So 92% of our comparisons, where we compared HPLC chlorophyll A and a fluorometric chlorophyll A, came out good. And it's really great. It has almost no bias, and we're really happy. But there's these dots that fall on the side, and we really want to know what's happening here. And going back to the first slide that you guys don't remember, but I said in the beginning that chlorophyll B also fluoresces in solvent. Some degraded pigments also fluoresce in solvent. So when you're doing the measurement of chlorophyll fluorescence, as you're going to learn today, you have to account for those. And sometimes we don't manage to account for all of them. And that's where that little distribution around the green good area happens. It can be also user error. This is taken from many, many people. But we managed to connect at least this portion down here with the areas where we know that there's lots of chlorophyll B. Okay, so guys are going to be doing that today. Okay, we're going to be extracting chlorophyll from the cells, putting it in acetone, waiting a little bit shorter than we would like, but that's great, and measure that chlorophyll. What you're going to be doing, all of you, you're going to be accounting for uncertainties as you're filtering, as you're collecting water, as you're extracting. And what we really want to see in the end is your final chlorophyll concentration, and you analyzing the sources of uncertainty. So putting those, like error bars that we're asking you, but also do the analysis of do an uncertainty budget. Try to figure out where the majority of that uncertainty is coming from. Is it coming from your measurements of volume? Is it coming from you spilling stuff and so on? It's going to be in your handout, okay? 7 minutes I have to go. Okay, so this is in solvent. This is a molecule in solvent. This is chemical process. However, then biology comes in. And this is where everything gets messed up. Chlorophyll in natural environment does not float happily in a cell in acetone. No, it doesn't. We spoke about that stuff. It lives somewhere. It lives inside of the chloroplast. Sasha's talked about that stuff. Or it lives inside of some protein complex, maybe not in chloroplast or in membranes of cyanobacteria, no? So if you're measuring chlorophyll in vivo, that's how it looks like. It's sitting inside of these complexes. And in eukaryotic cells, so in real cells, you have chloroplasts, and then you have these different structures. They're called thylakoid membranes, the stromas. And within these membranes, this is where these protein complexes, they're hugging our chlorophylls are living in, no? Inside the bacteria, they don't have organelles. So everything is happening in a membrane and these phycobilisomes. And I'm not going to go in detail, but just imagine that everything that is sitting in the membranes of these, something similar is sitting in the membrane of the cell of the cyanobacteria. How does the magic work? All right. So inside a membrane, first you have this, like, imagine like a gigantic antenna. And in this antenna, it's a protein complex in which you have a bunch of chlorophylls. But you have a bunch of other pigment helpers that Sasha was talking about. So what you have is all these chlorophyll and all these pigment helpers are capturing this beautiful, beautiful energy that's coming from the sun, no? And they're shuffling it back and forth, down the line all the way to the electron. But it's really good because you have different types of pigments. Chlorophyll likes to absorb at 470, for example, and then you have certain other pigments that like to absorb at 500 and so on and so on. So they're covering pretty good range of this wavelength spectrum, so they can capture lots of light. And since they overlap a bit, they don't lose any of that energy as they're shuffling it around. It's like, like supersonic MBA, like people, like really like ball is not lost ever. And then they dump this energy into the electron receptor, no? So now we're getting this different similar protein complex called prophotosystem. This is how these photosynthesis looks like, and they're insane, but somewhere in there, this insanity of the proteins, you actually have chlorophylls and some phyllophytins sitting. These are the really important ones because they receive so this whole insanity here is sitting in a membrane now, and they receive all that light. Yes, go ahead. So this is one photosystem too. Inside of the membrane, you have tons of them, no? So inside of the membrane of your chloroplast, you have a bunch of them. The photosystem too is usually housed around a couple of these light harvesting complexes, and they're all going towards sorry, this is my stuff. I have to check it. So yeah, photosystem two this is one. It's surrounded by these light like antennas, these light harvesting complexes. But there's a bunch of them, not just one. There's a zillion of them, and their number, because it's proportional to the need that the plant has to harvest the light. If you take the plant, take the phytoplankton and put it in darker light, it's going to start upping up the number of the amount of chlorophyll, probably increase the number of photosystems. The number of photosystems is dependent on the number of chloroplasts they have, and so on. So this is just a teeny, there's like a gazillion of these. Maybe not a gazillion, but there's lots of them. So just a protein complex that is there to accept that energy that's been now harvested by the light system. That energy gets taken to photosystem one, and this little electron, super happy electron, super excited, gets down the line to this whole system of little helpers into photosystem one. Photosystem two is now super sad and needs that electron. It's really excited to split the water, get electron back. This electron here is going to be shot down into the physiological processes of the cell that are responsible for the carbon sequestration. In theory, many different processes allow the conversion of the CO2 into the carbohydrates. So what does all this stuff has to do with the fluorescence? Going back to the Jablonski diagram that I wasn't really explaining too much, the energy that is absorbed by the molecule, in this case, molecules that are sitting in the cells, can be spent in 3 different ways. It can be used for photochemistry, photosynthesis. That's that little electron that went down the line, no? It can go into the vibrational modes of a molecule or heat, or it can be re-emitted as a fluorescence. So think about now sunlight shooting at these little, like, these antennas and stuff, and there's lots of sun, and now you're increasing the sunlight. They're like, yeah, yippee, yippee, and they're shooting all this, like, light and all this energy down the line towards the carbon cycle, no? And eventually that photosystem one and everything that's happening down the line is going to kind of start getting kind of crashed. Is going to be too much stuff. And they're like, we cannot accept any more of that energy. So some of that energy is going to be pushed out as a heat. It normally happens, no? So they cannot just shove all that crazy energy they're shoving around towards the physiology of the cell, towards the photochemistry. Some of it's going to be shoved out as a heat. If you start increasing the light, more of that energy is going to be shoved down as a heat. Cell likes to control the relationship of these three things. I think I have it written here, and actually can regulate the ratio of that energy that is distributed between the heat, photochemistry, and fluorescence. If you start turning one off, the energy that is absorbed has to go somewhere. It's going to go into heat or fluorescence. If you turn off the photosynthesis, the first photosynthesis. If you turn off, every time you turn off different steps, that energy has to be distributed somewhere, okay? There's certain things that can happen with the cell. If you have too much light being absorbed and the photosystems don't want to accept any more of that energy, you can have something that is called non-photochemical quenching, where a majority of that energy is going to go into that heat that I mentioned, no? But cell really likes to function a lot. So sometimes it can quench that heat process in order to take more energy towards the photosynthesis and this photochemistry, just photosynthesis project. No, so that's called, I mean, I should not go in this because it doesn't matter. The ratio between the energy that is distributed between the heat and photosynthesis and fluorescence, it's not constant. That's important stuff to really understand. So from that perspective, the amount of light that is absorbed by the cell versus the amount of light that's going to be fluoresced by the cell, it's not a constant thing. It's highly dependent on a happiness, capabilities, light, nutrient, many different things that are attacking that cell. So it is not a constant thing as before. So when you go back to the same thing where you're trying to measure fluorescence from the cell now, you have, let's say, so you're trying to solve, so you're still shooting the same light into the cell, you're measuring emission of the same light, you know that there's chlorophyll, you know that there's absorption. Will you be able to correlate these things? Will you be able to make a comparison between the fluorescence of the fluorescence of the chlorophyll and chlorophyll concentration? I said that this quantum yield of fluorescence, it's very variable in the cell. It depends on many different things. That's not the only things that is variable. When we have different types of cells, we have different types of pigments. It's not only chlorophyll that helps absorb that light. I said there's many different players in that absorption light process. So if you're trying to look at the excitation and emission spectra of different phytoplankton types, and all these colors of different phytoplankton types, if you try to excite them at different wavelengths and look at their emission in one specific wavelength, you get completely different curves. So this lambda 1 and lambda 2, and the quantum yield of fluorescence is going to vary a lot for different type of phytoplankton. We spoke, and actually Sasha spoke a little bit about absorption with the phytoplankton, or absorption of the phytoplankton star, where you have absorption of phytoplankton is divided by the chlorophyll. That's pretty much this term here, now. And here's a plot from an amazing paper by Annick Bricot in 1995, where she actually compared chlorophyll concentration versus A5 star in a specific wavelength. And it's super highly diverse. So we can't make that assumption that we made in solvent, that we know something about the relationship between absorption and a chlorophyll. It's insane. It's all over the place because there's different phytoplankton, there are different pigment filters, different pigment helpers. The physiology is different between different types of cells. So we're knowing all that stuff. We know that this mass-specific absorption, the A chlorophyll star or A5 star, is highly variable based on the phytoplankton community structure, based on the packaging effect that she was talking about. The quantum yield of fluorescence is going to be highly variable because, once again, it's a function of community composition, but it's also a function of happiness of the cell, the physiological state, and so on. So when you start varying all these things, it's really hard to get a constant relationship between chlorophyll fluorescence and chlorophyll concentration in vivo, once your chlorophyll is in a cell. And that's what you guys are going to be doing today, okay? If you actually go in a field and try to do exactly the same thing, you measure the chlorophyll fluorescence in situ and you measure chlorophyll fluorescence in solvent. And you get relationships such as this one. It's not one-to-one line. It's all over the place, no? And this is something that I did some time ago. And the way that I tried to put it on the same line is that I built a nonlinear best fit function that took into consideration light, temperature, year, day, and depth. Because it took into consideration the amount of light. Because we said if there's too much light shine on a cell starts freaking out and it starts turning off its reaction center because it's going to get damaged, like bye-bye. So you can see that really beautifully here because this is chlorophyll fluorescence divided by chlorophyll as a function of light. You can see that the relationship goes like down. That's your non-photochemical quenching here. My depth, my temperature, and my year day probably were a good explanation for phytoplankton community structure or the nutrient stress or something. So because when I did this correction, I kind of made it line up better, but not so great because it's still scattering around that one-to-one line. But it's good enough, no? That's what I did. Something similar was done by Luke. Where's Luke? And he looked at exactly here, Harpswell sound this similar relationship. No, am I correct? So he looked at the extracted chlorophyll versus raw chlorophyll fluorescence, and that's what you guys are going to be doing today. Trying to figure out this relationship between extracted chlorophyll and chlorophyll fluorescence are going to be measured with the instrument, chlorophyll in vivo. Can we use chlorophyll fluorescence as a phytoplankton biomass? We usually talk about phytoplankton biomass not as chlorophyll, but maybe carbon. So in that point, we're going to be comparing chlorophyll fluorescence to carbon. I don't have carbon usually parallel to that one. So I'm going to say, okay, I'm going to compare chlorophyll fluorescence to backscattering because that's my kind of approximation for the carbon in the ocean. And you can see that doesn't really work really well. In this case, it was a function of community composition and physiological status of these cells. And it's not just me, many people have shown that it's highly variable, this relationship of chlorophyll fluorescence with the backscattering across different times of the year in the open ocean. It's an awesome paper by LaCour. So again, this is what you guys are going to be doing. You're going to be calibrating in handheld FLNTU, we call it FLBB, a fluorescent instrument that we can deploy on gliders. We can deploy it, on the buoys. It's measuring chlorophyll fluorescence in live cells. And you're going to be parallelly taking samples of chlorophyll and measuring the actual chlorophyll concentration using in vitro chlorophyll fluorescence. You're going to be making a dilution series. You're going to be measuring the dark counts. You're going to be measuring the chlorophyll fluorescence. So fluorometric chlorophyll, each of these dilutions, you're going to be measuring the chlorophyll fluorescence of each of these dilutions and try to make this relationship work, no? And then you're going to fit a line and you're going to try to find a conversion factor, a scale factor. It's going to allow you to take any of the measurements that you're collecting with that chlorophyll fluorometer now, that you can deploy on a glider and a buoy and convert it to chlorophyll. And we're going to see how bad is it going to look like or good. And that's it. I did it in 36 minutes. This was super fast. Didn't go into details. We're going to have a lab now, and please ask us more. If you want to know more about vibrational states, ladies there. If you want to know how to misuse chlorophyll fluorescence, I'm here. But yeah, that's it. Any questions? Questions? Million questions? We don't have to clap. Yeah. Okay. And yeah, questions there later.